Good afternoon, everyone. All right, we're going to start. I was told that today is the beginning of the recruiting season, and we're really happy to see all these new medical students who want to apply for residency program. And so welcome, and we hope you have a great experience here in Pacific Northwest. It's a beautiful place and a great place to train, so we hope that you consider us closely. It's my pleasure today to introduce the speaker. Dr. David Holtzman is the Andrew B. and Gretchen P. Jones Professor and Chairman of the Department of Neurology at Washington University in St. Louis. He did his medical school at Northwestern University and his residency at UCSF. He received a 2003 Potemkin Award for Alzheimer's disease research, as well as he gave the 2014 presidential plenary session at this year's AAN. Since arriving at Washington University 20 years ago, he's trained 45 grad students, postdoctoral fellows, and physician scientists. One of them was me. Dr. Holtzman was my PhD thesis committee chair, so that was really great. His lab focuses on the neurobiology of ApoE and its receptors, how ApoE, A-beta binding proteins, neuronal activity, sleep influences A-beta metabolism. His lab has expanded its focus, and it's also studying tau metabolism and its possible prion-like spread mechanism. His work has resulted in Eli Lilly making a humanized monoclonal antibody, which is being tested in humans right now. And he'll talk about his exciting work in developing anti-tau antibody that's about to enter a phase 1 trial as potential therapy against tauopathies. So please welcome Dr. Holtzman for his talk on potential roles of extracellular tau in development and spread of tauopathies, effects of anti-tau antibodies.
David, there's a ground rule here. We've got audiences far and wide, and they sometimes don't hear questions that could come from the audience. So,
okay.
And I'll kind of start waving my arms, if you could just briefly repeat the question.
Okay, is this volume okay? Good. Well, thanks, Bruce and Leo, for the invitation to come to the University of Washington. I've been to Seattle a few times over the years, but I've never had the chance to visit the university. So this is an exciting day for me to be here. And I know a number of people here, but I had a chance today to meet some people I had not met before. So this has really been a fun day. So what I'm going to talk about today is something that I really haven't worked on so much until the last 4 years or so. As Leo mentioned, most of my lab has been focusing on trying to understand what controls the metabolism of the amyloid beta peptide that is relevant in Alzheimer's disease. But a few years ago, we also started getting interested in tau. And so what I'll do today, I'll give a brief introduction. This is Neurology Grand Rounds, a little bit about Alzheimer's disease in general, but then I'll focus the talk on some of the work we've been doing on the tau protein. So in terms of disclosures, which we're obligated to do, what's relevant today to any of these disclosures is that one of the antibodies that I'm going to talk about at the end, we wrote a patent on that was licensed by Washington University to a company on the therapeutic use of anti-tau antibodies, the potential therapeutic use. Okay, so most of the people in the room probably are very aware of what dementia is, but it's a decline in memory and other cognitive abilities that is sufficient to impair your social and occupational functioning. And one of the main reasons that we're also interested in this is because the prevalence of dementia is incredibly high the older you get. So by over the age of 85, it's upwards of 50% or more. And Alzheimer's disease probably contributes to about 75% of people who develop dementia in the United States and Western countries. So the clinical features of Alzheimer's disease that we're all aware of is that there's a very, very gradual onset and progression. Typically, episodic memory is the first thing that's noticed that's wrong with individuals with the problem, although even early on, there's difficulty with executive dysfunctioning, just as early as the memory problems, if you really look for this. In addition, there are difficulties as the disease progresses with language, praxis, visual spatial dysfunction. And behavioral dysfunction is frequent early in the disease with apathy, although as the disease progresses, you can certainly see a variety of other problems. The course of disease is variable. Typically, the disease from clinical onset to death is anywhere from 8 to 12 years, but that can be much shorter than that. It can be much longer than that. Just thinking about it very simplistically, we often think of this as there's 4 stages of the disease: very mild, when people are still pretty independent; mild, where they could still be living in the community, but maybe with just a little help; moderate, where somebody needs significant help, but maybe not in a nursing home; and severe is where people need around-the-clock care. And based on the symptoms and signs of the disease, you can usually be pretty accurate about the diagnosis based on just the clinical phenomenology. I can remember when I was in medical school, I was taught that you really need to rule out the diagnosis of Alzheimer's disease. And I think that's, for the most part, not true. You can pretty much tell when somebody's developing it with a very careful, informant-based history and exam. But as we know, or seeing a lot now, now that we can use biomarkers to pick up who really has the underlying pathology or not, even a really, really good clinician will be wrong 10% of the time simply because there's other things that can phenocopy the disease. The 2 major types of disease to which many people at the University of Washington have contributed to, such as Tom and others, is that there's the early onset familial form of disease, which is very infrequent, but has taught us a lot about the science of the disease. And then there's a late onset form, the common form that we see most often, where there's a significant genetic component, but it's not autosomal dominant in the same way. And the 2 strongest risk factors for the common form of disease are certainly age and genetics. So we've known for a long time about the pathologies of the disease based on findings from Alzheimer and his colleagues and many others in the early 1900s, when they described the 2 major neuropathological features of the disease, plaques and tangles. So as you can see in this image, plaques are predominantly extracellular structures which are made up mostly of the amyloid beta peptide in an aggregated form. And neurofibrillar tangles contain aggregated forms of the tau protein. In addition to these pathological features, as the disease progresses, there's striking atrophy of the brain. There's also nerve cell loss and dysfunction as the disease progresses, and there's activation of the innate immune system. So looked at schematically, this just illustrates that the amyloid beta peptide is a normal product of all cells, but particularly it's made at higher levels by neurons, and it's released in a soluble form. It's normal. It doesn't seem to do anything bad. However, when it aggregates, it forms these amyloid plaques. Surrounding the plaques, you can see activation of the innate immune system with microglia and astrocytes being more activated than normal. And you can also see, as I said earlier, that you can see the tau protein, a microtubule-associated protein, aggregating inside cell bodies and dendrites in the disease. I would say when I was first in medical school, it wasn't really clear, because the genetics of the disease weren't worked out yet, whether these lesions were byproducts or secondary in the disease or primary. And I think personally the data is now completely overwhelming that these proteins that aggregate, especially A-beta and tau, are central to the disease. They're not the only thing going on in the disease, but they're central in participating in disease pathogenesis. So why do I say that? A lot of this is due to genetic studies, biochemical studies, human studies, that argue that the amyloid beta, which is derived from the amyloid precursor protein, as I mentioned, is a normal protein that's made all the time. However, the genes that cause early onset familial Alzheimer's disease generally do one thing in common. If you have a mutation in APP itself, just outside of the A-beta region, that results in overproduction either of all A-beta species or just selectively the longer forms of A-beta, 42 versus the shorter forms. And somehow it appears that when the protein is present at a higher level throughout one's life, because it's a protein that can aggregate, it's amyloidogenic, the higher the level of an amyloidogenic protein, the more likely it's going to aggregate earlier in life. I think that probably explains why most people with both APP mutations as well as mutations in presenilin 1 and 2, which are components of gamma secretase, which cleaves A beta from APP, it's probably the main reason why these people get disease. But that doesn't explain the whole disease. That's the early onset form of disease. And how does the whole disease occur? When this peptide aggregates, that may be necessary but not sufficient for Alzheimer's disease. There's other things that need to happen. In late onset disease, it's very clear that ApoE is the strongest genetic risk factor we know of for late onset disease. And it does not affect A beta production. It actually affects its clearance and aggregation and regulates this process in addition to other neurobiological properties. But once A beta aggregates, while it may itself have some toxicity, it may induce inflammation, it really appears that other things are necessary for the disease to progress. And one of the main things that appears necessary is for tau to go from its soluble form to an aggregated form, and particularly spread in some way from hippocampus and entorhinal cortex out into the neocortex. And certainly there's evidence now genetically and from other studies that if you just have mutations that cause tau to aggregate earlier in the disease or earlier in the brain than normal, you will get a neurological disease independent of the amyloid beta peptide, such as hereditary forms of frontotemporal dementia, PSP, CBD, etc. So I think this isn't the whole story of Alzheimer's disease, but I think it emphasizes that the tau protein is probably an executioner in some way of disease, especially when it moves out into the cortex, as more imaging studies are showing now, following people along during the disease. So one of the main things that's emerged over the last 20 years is the fact that Alzheimer's disease doesn't start when you become symptomatic, not a big surprise. And a lot of longitudinal and cohort biomarker studies and I'll just summarize what I think. I'll interpret many of them to say that it looks like one of the first things that you can pick up that's abnormal related to Alzheimer's disease is that the amyloid beta peptide begins to aggregate in the brain about 15 years prior to any cognitive symptom or sign of disease. And as it builds up in the brain, people remain cognitively normal for quite a while. But at some point, one also sees neurodegeneration starting to occur as marked by neurofibrillary tangles, as well as markers of neurodegeneration either by neuroimaging or in the CSF that you can pick up. And once that starts to happen, that still is probably several years before symptoms begin. And if you follow people with these markers over time, they go on to develop dementia once this happens, but it occurs in some sort of sequential series of events as I'm showing here. So one of the things that's really, I think, going to be important and is occurring now is to identify people in this so-called preclinical stage of Alzheimer's disease before there is significant neuronal death going on so that one could try to treat the disease at that point. But in terms of this talk, I'm going to talk about tau, and I think there's a lot of evidence that as the disease as the symptomatic portion of the disease starts to occur, that's when the aggregated forms of tau move from just being in the hippocampus, which occurs in almost everybody as they get older, out into the neocortex. And once that happens, that seems to correlate with the progression of the symptoms of the disease. So focusing now on tau, what are its potential contributions? There's a variety of, many of the neurodegenerative disorders are proteinopathies, where proteins aggregate in the brain that normally don't aggregate. Tau can aggregate in neurons and in glia, depending on the disease. So it's present. Tauopathies include Alzheimer's disease, FTD, PSP, corticobasal degeneration. There's acquired forms of tauopathy, chronic traumatic encephalopathy. And in Alzheimer's disease, what appears to happen is that, based on Brock and Brock staging and many other studies, is that tau occurs in normal aging to aggregate in the hippocampus and a little bit in the entorhinal cortex. But in the course of Alzheimer's disease, it tends to spread out into parts of the temporal and frontal cortex and then out into many other parts of the cortex. And it's been theorized over the last several years that one of the ways this might occur is that it might occur in a prion-like fashion. So namely that it's spread somehow from cell to cell, so that when an aggregated form of the protein somehow gets to another cell, it induces aggregation in adjacent cells or synaptically connected cells. So this is still a hypothesis. It's not proved, but we'll talk about what is more the evidence for this and can one study this. So what I'm going to do is talk about studies that we've done in my lab trying to understand the metabolism not of intracellular tau, which is where it's mostly present, but extracellular tau. So we've been doing some studies to look at its metabolism in the extracellular space and determine a little bit more about how that may modulate pathogenesis. Then I'll talk about a study we published recently looking at how synaptic activity itself can regulate the release of tau. And then finally, some studies we've been doing on anti-tau antibodies and their potential therapeutic use. So as I was mentioning, tau appears to play a very important role not only in Alzheimer's disease, but in certain other diseases that appear to be relatively pure tauopathies. So certain forms of frontotemporal dementia are relatively pure tauopathies. PSP, CVD, or probably PSP is the most pure tauopathy where you don't see much other pathology other than tau. And then TBI, some forms of CTE appear to be relatively pure tauopathies. So I think we have a pretty poor understanding of the metabolism of this protein in vivo. As I mentioned, it's predominantly a cytosolic protein. It's microtubule associated, and it's clearly involved in regulating microtubules. But it's also found in the extracellular space. It's in the CSF normally, even without damaging the brain. And it's elevated in some tauopathies as disease progresses, particularly in Alzheimer's disease. But it's not elevated in the CSF in other tauopathies, such as FTD or PSP. So it's possible that the extracellular form of tau may be involved in the spreading of tau aggregation from cell to cell. That's, again, not proved, but there's evidence to suggest that might be the case. So when we came into this, my lab had been studying protein metabolism in the extracellular space of the brain using microdialysis, and I'll show you that in a moment. So we thought, well, one of my collaborators had just moved to Washington University who was studying the potential prion-like behavior of tau, who's Mark Diamond. And when he came to WashU, I started thinking about what we were doing to study protein metabolism in vivo in the brain. And with some of his studies suggesting that tau might spread from cell to cell, we started to develop further techniques to study tau outside of cells in the brain. So early on in around 2002, 2003, one of my graduate students at that time, John Cerrito, specifically developed a way to use microdialysis to study proteins in the extracellular space of the brain. Of course, microdialysis is a very commonly used technique to measure neurotransmitters and small molecules, but it's not easy to measure proteins because they're big and you have to use a semi-permeable membrane. You have to have things that cross it. But suffice it to say, if you use large pore membrane sizes, you can measure proteins, and we developed very sensitive ELISA techniques to measure quantitatively A-beta. And we published a number of papers where we can measure the A-beta peptide as frequently as every 30 minutes in a living animal or in humans. So we adapted this technique to see if we could measure tau in the extracellular space of the brain using microdialysis. And so to first prove that this would work, we simply took a test tube and put tau in the test tube and put a microdialysis probe in and said, does it get across the membrane? So we first started using 100 kilodalton size semi-permeable membranes. And when you first start doing microdialysis, what you can do is you can vary the flow rate of the fluid going through the probe. And this is called a zero flow method. And when you do that and you cause the fluid to go slower through the probe, you recover more protein. And we could show that we could measure tau in a test tube by microdialysis. And so once we were able to do that, we said, all right, well, let's go in vivo and see if we could measure tau in the extracellular space of the brain. So in order to do that, we said, all right, well, we can do this in wild-type mice, but we wanted to also use mice that express human tau and also mice that develop a tauopathy in the brain. So we began to use these mice called PS19 or P301S mice that Virginia Lee's lab had developed at the University of Pennsylvania. They overexpress the P301S mutation form of tau that causes a form of frontotemporal dementia. These mice, when they're up until about 5 months of age, the brain looks pretty normal. But after that age, they begin to develop tau aggregation in the cortex and the hippocampus, amygdala. As you can see here, this is stained with an antibody called AT8, which marks a hyperphosphorylated aggregated form of tau. You can also see that they develop classic tangle-like structures with silver stains and thioflavin S. And importantly, the mice also, as they get older, actually develop atrophy of the hippocampus and cortex. So unlike some of the mouse models to study amyloid deposition, which develop some features of Alzheimer's disease, those mice don't develop neuronal cell death or marked loss of synapses, whereas these mice actually get profound neurodegeneration as they get older. So the first thing we want to do is just show that we had tau ELISAs that specifically could measure human and mouse tau, and we were able to do that. And then we put microdialysis probes in the brain of mice and let them wake up and walk around and behave. And we did the same kind of experiment I just showed you, where we varied the flow rate of the microdialysate. And by doing that, you could measure the higher and higher concentration of tau. And you can then extrapolate back to ask how much tau is present in the extracellular space of the brain, called E-tau, or extrapolated concentration, extracellular concentration. And what we could find is that the concentration of human tau in the P301S mouse extracellular space was about 250 nanograms per ml, and in wild-type mice, it was 50 nanograms per ml. So we wondered, well, is that rational? So we knew actually from previous work that the P301S mouse model overexpresses human tau 5 times more than wild type. So this is exactly what we would have predicted, just based on was the ratio of the levels about what we would have seen. The one thing that was a little bit strange is that the concentration of tau in the CSF is much lower than what we would measure here. So for example, in human CSF, it's 500 picograms per ml or 250 picograms per ml, and in these mice, it's about a nanogram per ml. So it's much, much higher in the interstitial fluid, and this might be because once it's released from cells, it gets right back up into cells, or it sticks to the extracellular matrix and doesn't get out into the CSF. That's one possibility. This is just to show you the measurement, where we extrapolate that the measurement's about 250 nanograms per ml in the transgenic mice and 50 nanograms per ml in the wild-type mice. And when we do the experiment in tau knockout mice, we detect no tau, which is good.
Does it matter what part of the brain microdialysis is?
I suspect it would, but we haven't done that many experiments outside of the hippocampus for doing these measurements for microdialysis. We've done this for amyloid beta peptide, actually, and depending on where you put the probes, you can see very different levels. And we think part of that is due to differences in synaptic activity between different brain regions, because we know now that the A-beta peptide, its release from neurons, is regulated by synaptic activity. So we should go back and do that now. We haven't done that for tau. So we want to know what form of tau is present in the extracellular space. So if you just take brain lysates, we can see in a wild-type mouse and a Western blot, we can detect mouse tau. In the P301S mice, we can detect human and mouse tau. And then when we take the interstitial fluid and we immunoprecipitate tau from the interstitial fluid with different antibodies, we can find that there is both human and mouse tau that appears relatively full length in the interstitial fluid. For some reason, the human tau, despite in the brain lysates being bigger than mouse tau, it appears a little bit smaller than mouse tau in the interstitial fluid. And I don't know whether there's some truncation or other explanation, other post-translational modification of the molecule. So one of the things we began to do, because we had a model that developed tauopathy, was to study the levels of tau that occurred over time as the mice developed pathology. So what we found is that as the mice got older, and we measured tau in the interstitial fluid. Let me go back to this bottom lower panel. Is as the mice got to 6 months, right as they started to develop pathology, tau started to go down in the extracellular space. And that's despite the fact that there's accumulation of tau starting to happen inside neurons. And then as the mice got older, there was a big decline in tau that was present in the extracellular space. So we wondered, why is that happening? Is that due to cell death? Is that due to where we thought it might go up with cell death? Or is it possible that there's some equilibrium between the extracellular form of tau and the aggregated form such that it might be sequestering the extracellular form of the molecule? That appears to be what happens with the amyloid peptide. So one of the things we did is we just looked at, and this has been done before, we looked at brain lysates from these mice and looked at the soluble and insoluble forms of tau. So what you can see is in these transgenic mice, as they got to 12 months of age, the salt-soluble forms or the detergent-soluble forms, if anything, appeared to decrease a little bit. And the detergent insoluble form of tau was increasing, as has been seen previously. So we were thinking that maybe this is some sort of a sequestration of extracellular tau that might be happening, although again, most of the tau is inside the cell, not outside the cell. But just to get an idea, is it possible that if there were some extracellular aggregates present, might that lower what we measure as interstitial fluid tau? So to get at that, collaborating with Mark Diamond's lab, we took some forms of tau that just contained the microtubule binding domain. So that's the part of the molecule that allows it to really aggregate. So what we did was we infused this microtubule binding repeat domain into the extracellular space while doing microdialysis in a mouse. And what we found is that just over a few hours after doing that, what we measured of soluble tau in the extracellular space decreased. So we thought this was evidence, not proof, but evidence that maybe when there's an aggregated form present extracellularly, that it could sequester the soluble form of tau, and it would make it appear that it was actually going down, maybe because of sequestration. Okay, so the model that we proposed a few years ago was maybe there's extracellular tau. We don't know how it gets out of cells, but it's getting out normally. And that if there were extracellular or intracellular aggregates, maybe this tau would be sequestered. And if it's present, if these extracellular aggregates were really present, maybe that's a species that might be able to be transmitted from cell to cell. Although we certainly didn't show any of that in this work. So the next thing we began to work on was, well, what if tau might really spread from cell to cell, what might regulate its release from cells? So we began to look at whether synaptic activity itself might regulate. Let me fix this. Whether synaptic activity might affect the extracellular level of tau. So these experiments were done by a talented postdoc, Kaoru Yamada, and these were all done in vivo in awake behaving mice. And one of the powerful things about microdialysis is that you can infuse substances into the microdialysis probe by reverse microdialysis, like drugs, and you'll get effects immediately surrounding the probe to investigate whatever it is you're measuring. So, for example, in this experiment, Kaoru took a simply infused high potassium and then measured glucose. So glucose goes down because high potassium causes more activity. It caused lactate to go up, consistent with more neuronal activity. And over the same type of time frame, we can measure tau about every hour. It went up and actually stayed up for a few hours before dropping back down. When we first started finding this, I wondered, well, why is the time course of this, why is the elevation of tau lasting longer than the elevation of activity? And one of the reasons, as I'll show you in a moment for this, is that it turns out that the half-life of tau is actually pretty long. It's not hours, it's like weeks. And the effect we're probably seeing is from tau being released, but the effect that we're causing is only by reverse microdialysis, so it's right around the probe. So it could simply, after we see the elevation, it can just diffuse away to the other areas of the brain where this activity is not occurring. I think that's what explains this. So some of the other evidence that neuronal activity is involved in the release of tau, at least elevated activity, is that we give a GABA A receptor antagonist, picrotoxin. You can see by EEG that causes an increase in activity. You can see that when you do that, you can see that glucose goes down again, that lactate goes up, and that, again, tau goes up for the time that we're infusing the picrotoxin. Again, consistent with elevated neuronal activity causing a release of tau. So one of the things that we began to be asked was, well, how do you know this isn't due to cell death? That when you're causing these elevations of activity, you're not just causing cells to die and release intracellular tau. So we did some experiments where, again, we gave picrotoxin, and you can see that tau goes up. But then we gave a stimulus that caused cell death. So then we gave pilocarpine, which causes seizures and a lot of cell death. And when you do that, tau goes way up. It goes like up about 12 times over just these changes in activity. And if we do something, for example, like measure LDH as a marker of cell death, when we just cause an increase in activity, that doesn't do anything to LDH like picrotoxin. However, when we give pilocarpine, LDH goes way up. So I think this is not just due to cell death. I think this is actually due to a change in activity. We didn't formally, I don't think we've still formally proved that, but I think this is most consistent with this data. The other things we've looked at are NMDA. So when we give NMDA, you can cause an increase in activity. And if you do this at even with relatively low levels of NMDA that cause just a relatively subtle increase in activity, again, you get about a twofold increase in tau in the extracellular space. This can also be seen if you give, if you stimulate different glutamate receptors presynaptically, you can also see an elevation of tau release.
And this extracellular space that you're measuring, David, is cortex, right?
This is in hippocampus.
Hippocampus.
Right. We have done this experiment just a little bit in cortex, and you can see the same kind of an effect. So one of the things we're a little bit surprised about is we said, all right, well, let's do the opposite experiment. Let's decrease neuronal activity. So we infused tetrodotoxin, which obviously blocks sodium channels. It blocks action potentials. And when we did that, there was no change in tau. So we thought it might go down. However, if we give NMDA, we can see an increase in tau. And if we give NMDA in the presence of tetrodotoxin, that blocks the increase. So it seems like if we elevate activity, you can get an increase. If we block the elevated activity, you can block the effect. But for some reason, when we decrease activity, that does not decrease tau. So why might that be? And I think the reason for this is shown in the next slide. So it turns out that the half-life of both intracellular tau and extracellular tau is very long. So we did this experiment where we took mice that we obtained from the Mandelkau lab in Germany that you can turn off the expression of tau inducibly. So in these experiments, we basically took the mice, gave them tetracycline, tau expression turns off, but the mRNA turns off within 24 hours completely. And we followed the level of the protein. So if you look in the hippocampus, you can see that tau eventually goes down, but the half-life is about 12 days. If you do the same thing and measure tau in the extracellular space, that by microdialysis, you see the same effect. Basically, the half-life that we measure is estimated to be 11 days. So you can imagine if you suddenly decreased something that, if neuronal activity decreased and that was regulating tau, if you turn down tau, it's going to take a long time to see that change. However, if you cause something to cause a release of it, you could see that immediately. And I think that's why we see these differences between elevating activity seeing an increase and decreasing activity, we don't see a decrease. That's what we think so far. And I think there's a lot more work that needs to be done on this because this, like most cytoplasmic proteins, we have no idea how a protein like this gets out of the cell. You can see evidence of both every single astrocytic protein that's cytosolic and neuronal protein that's cytosolic outside of cells. You just find them in the CSF, you find them in the ISF, but what's regulating that process if it is not clear? It may not be regulated secretion, but the kind of things that the normal brain is doing all the time is somehow appears to be regulating the release of these proteins. So if this was just a protein that didn't really matter if it was outside the cell, this probably wouldn't be important. But it's possible that because of this whole idea that the aggregated forms of these proteins may actually spread throughout the brain, maybe it is important. Why is it that in Alzheimer's disease, when you start getting tauopathy in one part of the brain, do you see it start then aggregating in other synaptically connected parts of the brain? Why in PSP do you see certain networks where tau aggregates, it always goes within the same networks? Maybe this might be related to that, I don't know. So the last thing I'll talk about today is the effects of certain anti-tau antibodies on tauopathy in vivo, looking first at central effects and then peripheral effects. So there's a number of studies that have been published over the last, say, 6 or 7 years in which people have taken aggregated forms of tau and actively immunized mice, or they've taken monoclonal antibodies to tau and they've administered to mice that develop tauopathy. And I'm showing, most of these studies show some protective effects of doing that, either a decrease in pathology by immunostaining, sometimes they show improved behavior. And I must say, when I first saw these studies being published, I wasn't working at this time on tau. I said, why would this show any effect? Tau is a cytoplasmic protein. Antibodies don't generally get into cells. Why would you see an effect of this kind of treatment? So then this whole idea that perhaps there's an extracellular phase that these proteins can get out of cells. Maybe they're involved in a prion-like behavior. I said, well, maybe there's something to this. And these are some of the papers that then were published showing that, suggesting that tau, when it aggregates, might actually spread from one part of the brain to another part of the brain to another part of the brain. This is all mostly work in animals. So I began to wonder and started to think about doing experiments with my colleague Mark Diamond, where is it possible that aggregated forms of tau escape cells, get into other cells, whether they're synaptically connected, for example, or not, and corrupt the tau in those cells? Well, if that happens, and there's an extracellular phase to that phenomenon, one would predict that antibodies might be able to block that. So one of the ways that we first began to approach this is that my lab made a whole panel of antibodies against tau, monoclonal antibodies. And we made them initially just to study the tau protein, like being able to measure it quantitatively, do Western blots, do ELISAs, etc. But we took our panel of antibodies, and Mark Diamond's lab had made these cellular assays where you could measure the ability of tau aggregates to cause extracellular aggregates, to cause seeding of intracellular tau. So the principle of this assay is that they would take tau and label it with one fluorescent probe, take tau and label it with another fluorescent probe, and if the tau actually comes together, you'll get FRET, or fluorescence resonance energy transfer. So this is a nice assay that their lab developed, where again, if tau gets inside the cell, it will cause aggregation in some way of tau that's in the cell, and you could detect this with FRET, and this could be done in a high throughput fashion. So we took this assay that Mark had developed, and we tested some of the antibodies we had developed just in cell culture. And what we found is that some of the antibodies, as shown in the left panel, were able to block the ability of tau to aggregate inside of cells, and some were not. And what this is showing is we took aggregates from brain lysase from these mice that developed tauopathy. We put them on the cells. You can see that they show strong FRET response, shown in this left panel. And in the presence of some antibodies, this one in particular, there was really strong blockade of the ability of that aggregate to induce FRET inside the cell. So, and this is just a dose-response curve with those different antibodies. So we decided, all right, there are some papers published on this, but really hadn't looked so much at the mechanism of how these antibodies might work. Let's take some of these antibodies, put them in the brain, in the ventricle of animals over time, where we know they get into the brain and be present, and see what they do to some of these animal models that get tauopathy. This is just to show you with one of our newer assays that Mark's lab developed, that if we take this particular antibody called 8.5, that you can take a lysate, it will induce FRET really dramatically, and if you deplete tau with this antibody, you can block the ability of the lysate to cause aggregation. And this doesn't work if you use an irrelevant antibody that's to another molecule. So to just show you the property of some of the antibodies that we made, if you focus on the bottom, we were able to make some very, very high affinity antibodies to both monomeric and aggregated tau. So this antibody called 8.5, which I'll talk more about, this has a KD of tau of 0.3 picomolar. It's specific only for human tau. It doesn't recognize mouse tau. This particular antibody, 9.3, has a pretty high affinity also, 99 picomolar, and 9.4, which was made to mouse tau but also sees human tau, has an affinity of 6.9 picomolar. And each of these antibodies is to a different epitope of tau, but they do not detect a phosphorylated epitope of tau. So if you take, just as an example, this particular antibody, 8.5, and you simply do immunostaining of a brain, you can see that it doesn't stain a tau knockout brain. It doesn't stain a wild-type mouse brain because this antibody is specific for human tau. It strongly stains a P3ONS brain without any tau pathology when it's young. And that's because tau is throughout the neuropil. It's throughout the cell bodies and the dendrites and or in the axons. And then as the mice develop tangle-like pathology, it stains not only the monomeric form of tau in the neuropil, it also stains neurofibrillary tangles. We use the same antibody to stain an AD brain, it stains neurofibrillary tangles. So this particular antibody stains both soluble tau as well as aggregated tau. So we characterized these a little further, and again, we were able to find that you could immunoprecipitate away the form of tau from brain lysates that's able to seed tau in these cellular assays. And if you immunoprecipitate tau from brain lysates from these transgenic animals that develop tauopathy, even the soluble fraction of the brain has not only monomeric forms of tau, but a whole smear of aggregated oligomeric forms of tau that these antibodies are able to recognize. And if you isolate these material that you immunoprecipitate either from an AD brain or from a P3ONS mouse brain, you can see that you can pull out these oligomeric structures, which in this case, this antibody can pull out fibrils, it can pull out these small oligomeric structures of tau. So whether these forms of tau are present only inside cells or outside cells, this doesn't tell you that, but at least we know these antibodies can recognize both soluble monomeric forms as well as aggregated forms of the molecule. So the first in vivo studies we did, as I mentioned earlier, we actually took mini osmotic pumps and infused tau in the ventricle of P3ONS mice starting at 6 months of age. So this is right when this particular animal model starts to get tau aggregation in the brain. And we actually infused the antibodies for 3 months. We had to change the pump one time in the middle so that the infusion would last the whole 3 months. And we infused, you can see 7.2 micrograms of antibody per day. And we picked these doses of antibodies based on other studies that have been done actually in the A-beta immunotherapy field. So what we saw as an outcome at 9 months, we did a variety of things, but we found very striking findings looking at the tau pathology. So this mouse here is an animal at 9 months of age stained with an antibody to tau called AT8 that stains, again, a hyperphosphorylated form of tau that's present in tau aggregates. And you can see throughout the piriform cortex and amygdala, there's a lot of abnormal tau staining. And that's true also when we infuse an animal with a control anti-A-beta antibody, which doesn't have any effect on this. But with the different tau antibodies, this one in particular called 8.5, we saw a very marked reduction in the amount of abnormal AT8 staining in the brain. This is quantified here, where if we look in multiple regions of the brain, piriform cortex, entorhinal cortex, amygdala, there's a very significant decrease in the amount of abnormal tau staining in these brain areas. There's also the other antibodies had some effect as well. It just wasn't quite as strong as this HG8.5 antibody. This is to show that when we look at the different forms of tau biochemically, the only form that we see a difference in when we extract the brain is the insoluble form. So if we look at tau that's present in these different treated groups that is salt soluble, detergent soluble, there's no difference between the amount of tau in the brain. But what we find a difference in is the amount of detergent insoluble tau, where there's more than a 50% decrease with 2 of the antibodies that we treated the animals with. We can also see an effect on sort of more classic markers of fibrillar tau. If you stain an amyloidogenic protein like tau, when it's aggregated, you can see strong staining in these P3ONS mice in certain parts of the brain. And in the animals treated with the different tau antibodies, there wasn't as strong as an effect as with the tau immunostaining, but there was about a 30 to 40% decrease in neurofibrillary tangles as defined by thioflavin S staining in the treated mice. The other thing that we looked at is microgliosis. At least it looked at quote-unquote activated microglia, in this case stained by the antibody CD68. And like most neurodegenerative diseases, if there is aggregation of abnormal proteins and there's damage, you see more staining with a marker like this. But in the animals that were treated with the anti-tau antibodies, there was less area covered by microglia as stained with this particular antibody. The other thing we did, which was, it's hard to do, it turns out, in these mice, is some behavioral tests. It's not hard to do the behavioral tests, but it turns out despite the fact that these mice are developing tangles and some brain atrophy, they actually don't decline very much in most of the tests that we gave them, including the water maze and other tests. However, as I'll show you in a minute, there are a few things that we could find abnormal, and when we do a test called conditioned fear, there were some abnormalities. So what you do is you basically put the mouse in a chamber, you give the animals a shock, and after they get shocks, they start to freeze. You then come back the next day and you put them back in the same chamber. You don't give them a shock, but if they remember where they were and they received the shock, they will freeze. And that's an amygdala hippocampal dependent process. And what you can see is in these bottom 2 graphs, in the bottom line in particular, on the second day when we put the animals that were treated with PBS in, they froze very little in the chamber. But the animals that were treated with 2 of the different antibodies, in particular the one treated with this antibody HJ8.5, they froze significantly more than the animals that were given the vehicle. So this implies perhaps that the antibody wasn't just decreasing pathology, that it might be improving the animal's function. I think it's tough with these assays unless there's really a number of them that are abnormal to come to a firm conclusion, but at least it supported the idea that the pathology reduction might be having a functional effect. Okay, so the last thing I'll show you today is evidence. Well, if you actually develop these antibodies as a therapy, one of the ways that you would want to try to administer them is, peripherally, without having to put a cannula in the brain. And we all know that antibodies don't get into the brain very well because they're big molecules. But about 0.1 to 0.2% of the concentration of an antibody in the blood is present in the brain because everything gets across to some extent. It just doesn't get across at high levels. So we basically made a calculation that by giving different doses of antibody peripherally, if we gave a certain dose, could we get the same amount of antibody in the extracellular space of the brain as we were administering ICV? And it turns out if we give about 50 milligrams per kilogram of this HJ8.5 antibody, the rough calculation is that we were getting about the same amount of antibody in the extracellular space of the brain as by giving it in the ventricle at the doses we gave, roughly. So we went and began to test that dose of antibody in exactly the same way as we just did with these ICV experiments, where we took the animals at 6 months, we gave them either a control like PBS, or we gave them this antibody at 50 milligrams per kilogram per week intraperitoneally, and then treated them for 3 months. So what we found is that at the dose of 50 milligrams per kilogram, again, there was no effect on the biochemistry of tau in the soluble fractions of the brain. But in the insoluble fraction of the brain that's detergent and soluble, that contains the neurofibrillary tangle-like material, there was a strong decrease of insoluble tau, at least with this higher dose of antibody. We also looked at immunostaining for tau, abnormally phosphorylated tau, again with AT8. And again, in some areas of the brain, such as the CA1 region of the hippocampus, we could see a significant decrease of p-tau as stained by this antibody after 3 months of treatment. We could also see a less strong effect, but a significant effect on thioflavin S staining, as shown here in the vehicle versus the HC8.5 treated animals. And then the one thing that we found, which hasn't been seen before to my knowledge, is that because these mice develop brain atrophy between 6 and 9 months of age, we determined whether the brain size was any different. And what we found is that first, if you look at 6-month-old mice, comparing them to wild-type mice, the size of the hippocampus and the size of the cortex is normal compared to a wild-type mouse. But between 6 and 9 months, there's about a 25% loss of cortical and hippocampal volume. And in the mice that were treated with antibody at both doses, there was about the hippocampus and cortex are about 5% larger than the mice treated with vehicle. This isn't blocking atrophy, it appears to be slowing down the atrophy. So I think of all these experiments, I'm not aware that in these immunotherapy experiments, people have yet looked or seen that the antibodies actually reduce something that is certainly consistent with it reducing the rate of degeneration. So I think if these kind of treatments ever get into people, this is certainly something that would be interesting to look at to see if you could actually block the rate of degeneration. And then the final thing we've done is it turns out that in addition to some behavioral abnormalities you can pick up in these mice, they actually also have decreased motor function as they get older. And so at the higher doses of antibody we gave on a test called the time on the screen, where you take you have the mice sitting on a screen and you invert the screen, they hang on. And the mice that are given antibody hang on longer, and also on a ledge test where they're put on into the ledge, they can stay longer on the ledge, implying that their motor sensory function is improved on the higher dose of the antibody. So how do these things work? We don't know 100%, but we've explored 2 potential mechanisms. So we've looked at the ability of in vitro of these kind of antibodies to either increase or decrease aggregated tau getting into immune cells like microglia. So what we find is if we take aggregated tau and we apply them to a microglia-like cell, a BV2 cell, in the presence of this antibody to tau HC8.5, you can see a significant increase in the ability of microglia to take up tau. Presumably this is through the Fc domain of the antibody. We do not see this effect with neurons. The other thing that we found, which is really striking, is that the level of tau in the blood of a human or an animal is very low. But after we've given antibody to these mice for 3 months, we looked in the blood, can we detect tau in the blood? There's actually a pretty strong increase in tau in the blood of the mice after 3 months that's dose dependent. And we've also looked to see, even when you give these antibodies acutely to mice and look just over a day or 2, you can also see a big increase of tau in the plasma. And it turns out if you immunodeplete the plasma of antibody, the tau goes away. So it looks like most of this tau is bound to antibody in the blood. So we're further exploring now what this is due to. We think one possibility is there's some tau that's getting out of the brain and the antibody is simply capturing it there and prolonging its half-life. But I think it's also likely that we know that antibodies cycle into and out of the brain, and so it's also possible that it's getting into the brain, capturing tau there, and then coming back out, and then we're measuring it in the blood. So we're really excited about this because this might be something to investigate further as a potential diagnostic test to see if we can measure tau now in the blood, and does it differ in the presence and the absence of tau pathology, or look at the effects of a treatment on this. So some of the next questions we're now beginning to ask are tau oligomers, or these tau seeds, actually present in the extracellular space of the brain? And so we're beginning to address this using microdialysis probes at a very large cutoff pore sizes, like 1 million Daltons, 3 million Daltons. Do anti-tau antibodies actually affect the level of tau in the extracellular space? We're just starting those experiments. And is the Fc domain of an antibody required for its effect to reduce tau pathology? So I'll just summarize what I talked about today. Tau is present in the extracellular space of the brain at pretty high concentrations. Tau oligomers may escape from brain cells, we don't know that yet, and enable spreading from cell to cell. And we're beginning to attempt to detect these tau seeds and whether they're really present in the extracellular space of the brain. We've seen that excitatory activity increases tau in vivo, and I think this potentially has important implications in regard to transsynaptic spread of tau within brain networks. And then finally, certain anti-tau antibodies given either centrally or peripherally appear to reduce tau pathology, and this approach, I think, has some promise to explore in human tauopathies as a therapy. So I think I thank the people that did most of these experiments along the way. I wanted to point out Hong Zhang, who made all the different tau antibodies in my lab over the last several years. Karo Yamada did the microdialysis experiments with synaptic activity, and Kiran Yanamandra did the anti-tau antibody experiments. And then finally, Mark Diamond has been a major collaborator for all the studies I talked to you about today. So thanks.
David, maybe I'll start out. I had a couple of questions. One of them, to start, just a clarification. Did I understand you correctly? You find a lot less extracellular tau in human CSF than you do in the extracellular space of mice.
No, so what we see is that the level of tau in the CSF of mice and in humans is lower than the concentration of tau in the extracellular space of the brain itself, like in the interstitial fluid space of the brain.
Got it. So it's not a, so it's equivalent.
I think it's probably not escaping from the interstitial fluid into the CSF as effectively as other molecules do. Maybe because it sticks to the to extracellular matrix or gets into cells once it's released. So there's more there than I would have anticipated based on CSF studies.
Got it. And then the repeating thing we're going to make you do. What about white matter? Mouse doesn't have a lot of white matter, so you're not going to get a good crack at it. But tell me what your thinking is about whether this pathology might in fact be importantly involved in white matter in addition to cortical area.
Right. So the question is tau or tau pathology relevant to the white matter in whether it's an animal or a human disease? So. Well, one thing to state, I didn't get into this because of time, I mean, there's some tauopathies where tau aggregates in white matter, in oligodendrocytes. Mostly it tends to aggregate in neurons in most of these diseases. But I think there's a lot of evidence that after axonal injury, that in areas where there is a lot of white matter, tau might be involved in the damage that is, or might be involved in axonal injury after, which is obviously in white matter. But this whole process of extracellular tau and whether that is relevant to axons or white matter or myelin, I have absolutely no idea.
Even if it's different, if the amount of tau in the extracellular space of white matter is dramatically different than it is in gray matter, that would be very
That would be good to, so is the amount of tau different between extracellular space of white versus gray? I would predict it would be very different, probably because if there's myelin around most of the axons in white matter, it probably wouldn't be able to get into the probe if it's stuck around right around the axon within the myelin sheath. But I don't know. I mean, we should be able to look and see. It might be tough to do in the mouse, as you say, because of the fact that white matter is not that big of an area. But maybe in a bigger animal, we could assess that. I would predict it to be a lot lower in the extracellular space in white matter than gray because of that. But I don't know if other people have tried to measure proteins by microdialysis in white matter versus gray matter before, to my knowledge. I'm not sure that's been done. You would probably know. I don't know if anyone's ever done that.
I don't.
Yeah. Yes?
Does this antibody circulate in the interstitial fluid? If so, how do you establish that? Fluorescent probes to look at.
Yeah, so so the question is, if there's an antibody present in the blood, how do you know what the concentration would be in the extracellular space of the brain? So the only thing we've done to assess that so far is we've administered antibody, like others have done, and then we've obtained CSF, both from, for example, from these mice. And what we can measure is that the concentration of antibody is what you would predict from many other studies people have done. It's about 0.15% of the concentration in the blood. So your question is, what's the concentration in the interstitial fluid space? And we've actually never done that. We're doing that right now to really determine whether that might be possible. And the reason we hadn't done it before is we hadn't been using microdialysis probes that were large enough pore size to let an antibody cross. But now we are using some, and so we're trying to directly measure that. So we've been doing some in vitro microdialysis experiments, and we now know we can definitely measure antibody in a solution that will cross the probe so that we can measure it. So we just started these experiments in the last few weeks to see if we can measure the antibody in the interstitial fluid space of the brain.
David, there's a question from outside.
Oh, okay.
What did I do?
Okay, so given that there are different forms of tau that are recognized by various antibodies, could there be multiple pools of tau that are released by neurons in different ways, i.e. an electrical activity-mediated release pool and a pool that is released in a slow, constant manner? That might explain in part the long half-life you measure when giving TTX, turning off the electrical activity-mediated release pool. I think that is something that is a very good point, and it's very possible. So no one had been doing this before, so now we need to go back and start determining whether that's the case. I think that's a really good question. Yeah.
So some of the highest CSF taus I've ever seen are in CJD.
Right.
But that's not thought of as a tauopathy.
Correct.
So what's going on?
So the one thing, yeah, so Dr. Bird is exactly correct. In CJD, it's probably one of the best markers of CJD in the CSF is the level of tau. It's not just elevated, it's really elevated. So the one thing is that many neuronal proteins, not all, but many cytosolic neuronal proteins, if you cause cell injury or death, they are released. So tau is one of those. It's not, I don't think it's specific for tau, but it's a very, it's easy to measure it with good ELISAs, and so I think the reason it goes so high in CJD is there's so much active neurodegeneration going on. You can see a big increase in tau after a stroke. You can even pick it up in the blood if you assay it early enough after the stroke for about 24 hours, then it starts going down. So I think that's a different phenomenon than what we're probably talking about here. Yeah.
So you demonstrated that with infusions of things like or toxin that induce seizure-like activity, you see this significant increase in tau. And I'm wondering, that would predict that if someone with Alzheimer's disease develops seizures, you might expect that their disease would accelerate in some way. Has that seen, and should we be looking closely to see if they were having seizures?
Right, that's a good question. So I don't know that I've ever seen a clinical study that has looked in humans at people that have AD who have seizures to know whether it actually accelerates the disease process or not. I mean, certainly anything that causes more neuronal dysfunction in a person that has Alzheimer's disease makes them worse if that keeps going on. But I don't know if there's any good studies suggesting that if you have seizures in AD that the pathology gets worse. But that might be hard to really look at just from human data unless you had a well-characterized cohort of people that had that versus not. And so, yes.
So there are tau PET scans coming along, and what do you think about that in the clinical realm?
Mm-hmm.
And would antibody treatment change the tau PET scans? Is that going to be a way to look at what you're doing?
Right. So one of the major advances in imaging Alzheimer's-like pathology in the last 2 years has been, and other tau pathology and tauopathies, is the ability to detect tau aggregation in living brain of humans with some of these new PET tracers. And some of the data that's been shown in the last year at different meetings and I know there's a paper or two that will come out in the next month on this. They look very impressive at being able to detect aggregated forms of tau in living humans and exactly where it's occurring. And I think that's going to be a major assist, in addition to the fluid biomarkers and amyloid imaging, to really say where is, what looks like so far that's been presented by people like Keith Johnson is that if you take people who have amyloidosis in the brain who are still normal, most of those people only have tau by that imaging agent in the hippocampus, a little bit in the entorhinal cortex. But in the people just before they are likely to get demented and people are very mildly demented, it looks like that's when you start seeing tau by imaging out into parts of the neocortex, temporal neocortex. And then as they get more advanced, you're seeing it. Again, it's limited numbers of people, but it looks like that so far. So I think that's going to be a great marker, not only as a biomarker for disease, but also if there's something like what I talked about today that really blocks that spread, that should be a perfect way to determine whether it's really doing that. And in fact, in Alzheimer's disease would be the best example because it really does appear to move around the brain in a relatively predictable fashion. So,
Can I ask one more
yeah.
question? I don't want to.
No,
Go.
right, go.
You show that nice graph, sort of a theoretical graph of the natural history of Alzheimer's disease, and amyloid going up first and then tau after that. So I think a $64 question is, are those two curves related?
Yeah.
And if so, how? Is the amyloid accumulation, does that have anything to do with subsequent tau elevation?
So the question is does the amyloid build up in the brain related to the buildup of tau in the brain? That's really right. And that, I think it absolutely is. I think we don't understand why. But the reason I say that is that just from all of the clinical pathological studies that have been done in humans, as well as some experimental work in animals, argues that while they don't overlap in where they occur, at least in humans, you don't start getting the pattern of tauopathy in the brain in humans that looks like Alzheimer's disease patterns until you also have amyloid in the brain. And there are certainly examples where you can get amyloid deposition where you're not yet getting that pattern, but then those are people generally that still are normal and haven't progressed on to develop clinical symptoms. And I think there's many other reasons why I think this, and I actually have written in sort of a review about why I think that. I can send it to you. But I think there is a lot of evidence that suggests it's driving the tauopathy. Maybe once the tauopathy starts to get to a certain point, it may be independent of that. And clearly there's other tauopathies where you don't need A beta to be around to move. But I think in this pattern of that you see in AD, it seems to accelerate that process. But yeah.
You mentioned the finding of behavioral or cognitive behavioral phenotype was a little bit difficult. You said that on the water gates and other bases they were relatively similar, but kind of only on the textual. Was that surprising?
Yeah, I mean, I think I don't know why. Why when you're losing cells in the brain, you're losing. You're clearly getting synaptic damage, axonal damage. Why the animals don't have more behavioral phenotype? I mean, there must maybe the areas there's enough reserve that they just don't have it. I mean, I don't know. I mean, but I think it's really mysterious. Some animals that don't have synaptic loss and don't have neuronal death can show behavioral abnormalities, presumably because whatever is going on in the brain pathologically is affecting synaptic function, but not affecting cell death enough to cause that.
At least at the level of CA1, your cellular findings are pretty.
Yeah. Well, we're not seeing. We may be seeing some cell loss, but most of the I was just showing you the tau pathology, not necessarily. There's not necessarily dramatic cell loss there. So, yeah. Yes.
David, you and I were chatting, and you mentioned that Micah Nettergaard had been by your place, and she's doing some exciting work about CSF percolating through the brain, cleaning things up at night. My question, and now you've got us thinking about extracellular tau, to the extent that this system is crucial, actually, let me just phrase it as a question. Do you think this system is crucial, perhaps, in setting tau levels? And you could perhaps address it initially by looking at light and dark.
Right. So one of the things that Micah Nettergaard in particular has been working on the last few years is what she terms the glymphatic system. So what I'm talking about in these studies is the extracellular space of the brain, the interstitial fluid, the so-called Burkow-Robin spaces of the brain. And Macon's work has recently shown that aquaporin 4 channels appear to regulate water content in the extracellular space of the brain, and that when animals are awake, that space is smaller, and then when they're asleep, it's larger. And there's evidence that she's presented and published that clearance of proteins from the brain is greater during sleep, possibly because the space is bigger and it allows transport of that fluid from ISF into CSF and into the lymph nodes in the head. I think this could be those observations could be very relevant to protein clearance in general in the brain, and also in these diseases. And I think the challenge now is to follow up her initial observations and really do different manipulations to determine how important that clearance is in these processes. That's what's not clear yet, is how relevant it will be to the disease process.
I think you've satisfied everybody, David.
Okay.
Thank you so much.